All right, so our first seminar today is by Dr. Thomas Steeger. He's a senior science advisor at in the Environmental Fate and Effects Division of EPA's Office of Pesticide Programs. Next slide. He received his PhD from Auburn University, where he studied the effects of environmental pollutants on hepatic vitalogenesis and receptor-mediated uptake of vitellogenin by oocytes and catfish. While completing his graduate studies, he also served as the vice president of a small consulting firm, which worked with State Department of Natural Resources in evaluating recreational freshwater fisheries across the Southeast United States. After completing his PhD, he went on to work for the U.S. Environmental Protection Agency, and during his 24 years with the EPA, he served as one of the lead technical advisors on the development of the EPA's guidance for assessing the risk of pesticide to bees, and on the development of the national strategy to promote the health of honeybees and other pollinators. He currently serves as a committee member, delegate, and representative of multiple EPA work groups focused on pollinator health and the effects of pesticides on pollinators. Next slide.
Okay, great. And the next and the last speaker for today will be Dr. Alessio Ippolito. He holds a master's degree in environmental science and a PhD in ecotoxicology from the University of Milano Bicocca. And his academic activities on the ecotoxicology of pesticide fed into several EU research projects. To mention a few, there are Alarm and No Miracle. And currently he works in the position of a scientific officer for the EFSA in the pesticide department. But before I come to his talks, here's just a small overview about his former career. So from 2012 to 2014, he worked as an expert of pesticide environmental fate and ecotoxicology in the International Center for Pesticides and Health Risk Prevention. And afterwards, 2014, he worked for the Institute for Environment and Sustainability at the Joint Research Center of the EU Commission, where he was dealing with a review of the priority substance list under the Water Framework Directive. And now, for 6 years now, he's working in the European Food Safety Authority, where he is dealing with the environmental risk assessment of pesticides. His activities include coordinating the EU peer review of pesticide active substances, performing ad hoc assessments, and developing risk assessment methodologies. In the last couple of years, he has been focusing on the revision of the risk assessment methods for bees and implementation of effect modeling and risk assessment. So that is also the topic for today. Great. And in the next context, and the context of his work, he authored about 50 between EFSA outputs, a technical report of the EU Commission, and papers published on peer-reviewed journals. Here's just a small overview about his published work, and most of them can be found in the internet and on the ResearchGate. And so much for the background of our experts. If you have any questions, feel free to post them into the chat. They will be answered after the lecture. So then I give the word to Thomas Steyer. Thank you.
All right. Thank you so much for the introductions, Jana and Katie. So you may have noticed that Tom is not here with us in person. That is because he actually sent me a pre-recorded talk that's going to introduce you to the framework that he uses for assessing risk to bees within the US EPA. So it's pre-recorded and it's pretty short. It's only about 10 minutes, and then we'll bring things over to Alessio. And so just hold tight, and I'm going to share my screen and we'll get this all up. Okay, so hopefully you can all see that.
I don't think we can hear it, Natalie. I think you might need to there's like an option to share computer audio on
Oh,
Zoom.
no. Oh, I can hear
Yeah.
it. Oh, no.
Yeah, I can't. I don't know if anybody else can,
All right.
but yeah, if you're like up in that upper menu bar for when you're sharing your screen and you go over to like the extra, there's like 3 dots, you can open
Oh,
that and then it says share computer audio.
got it. Okay, let's try this again.
Thank you for this opportunity to present on EPA's process for assessing risk of pesticides to bees. To assess the extent to which pesticides can potentially affect non-target organisms, EPA requires multiple studies on a range of taxa, for example, mammals, birds, fish, aquatic and terrestrial plants, and aquatic and terrestrial invertebrates. For aquatic and terrestrial animals, EPA requires acute and chronic toxicity tests. These testing requirements are specified in Title 40 of the Code of Federal Regulations, Part 158. With respect to bee-related testing, in 2011, EPA issued interim guidance on exposure and effects data on bees. The guidance was based on the agency's understanding of the evolving science through a 2011 Society of Environmental Toxicology and Chemistry Global Pelston Workshop on pollinator risk assessment, and by work underway in Europe through the European and Mediterranean Plant Protection Organization. In 2012, EPA, in collaboration with Health Canada's Pest Management Regulatory Agency and the California Department of Pesticide Regulation, presented a white paper to EPA's Federal Advisory Committee, the FIFRA Scientific Advisory Panel, describing a conceptual framework for assessing risks of pesticides to bees. Based on that white paper, input from the Scientific Advisory Panel, and additional guidance documents generated through the European Food Safety Authority, EPA, Pest Management Regulatory Agency of Canada, and California Department of Pesticide Regulation released the harmonized guidance for assessing pesticide risk to bees in 2014. The guidance has subsequently been translated into Spanish for consideration as a US, Mexico, and Canadian guidance document, and was considered by Environment Australia in their development of a roadmap for assessing risk of pesticides to bees. In addition to the 2014 guidance, in 2016, EPA issued a guidance document for agency risk assessors, which discusses the regulatory provisions for requiring data, the current pollinator data requirements for conventional pesticides, and additional bee toxicity and exposure studies. While EPA has been evaluating risk to bees for over 20 years by requiring data on adult bees and, when needed, on whole bee colonies, the new data requirements address uncertainties regarding not only acute contact exposure of adult bees, but now include adult acute oral toxicity following the Organization for Economic Cooperation and Development, abbreviated OECD, Test Guideline 213, and acute toxicity tests with larvae, OECD Test Guideline 237, as well as chronic toxicity tests with adult and larval bees. The chronic toxicity test with adult bees follows OECD Test Guideline 245 and examines young adult bees dosed for 10 consecutive days, while the chronic toxicity test with larval bees, OECD Guidance Document 239, doses larvae for 4 consecutive days until they enter pupation and stop feeding. This test extends through adult emergence and therefore represents the full development of the worker bee from egg to adult. In addition to monitoring for bee mortality, these tests must also report any sublethal effects that may occur during the study. Depending on the outcome of those tests and the risk manager need for further characterization, additional semi-field studies, for example, enclosure studies of colonies following OECD Guidance Document 75 or colony level feeding studies described by Uman, and full field colony studies according to EPA 850-3040 may be required. In addition to the colony level tests, exposure data may also be required on residues in pollen and nectar. Risk is commonly defined as the likelihood and magnitude of an adverse effect from exposure to a particular stressors, for example, a pesticide. Therefore, risk is a function of both the extent of exposure and the toxicity of the pesticide, and that toxicity can vary depending on the taxon and species tested. Risk assessments can be deterministic, that is, based on point estimates, or they can be probabilistic and based on a distribution of exposure and sensitivity estimates. Using a deterministic approach, EPA evaluates the ratio of point estimates of exposure to point estimates of toxicity in what is referred to as the risk quotient, which is abbreviated RQ. The agency then compares the risk quotient, RQ values, to agency levels of concern, which represent thresholds for mitigation. EPA relies on multiple lines of evidence in assessing risk. These include registrant-submitted exposure and effect studies, open literature studies that meet the standards specified by agency guidelines, and bee-kill incident reports. Bee-kill incident reports are submitted through the EPA beekill@epa.gov portal, the National Pesticide Information Center, and state and tribal lead agency investigations of incidents. This is a simplified depiction of the three-tiered process used by EPA to assess risk of pesticides to bees, where the answer to multiple questions determine the extent to which higher tier studies or refinements are needed. For example, the most basic question is whether it is likely that bees are exposed. This involves an understanding of whether the crop may be attractive to bees, whether its application occurs at or near a bloom, and the time at which compounds are applied. Also, transition to higher levels of refinement is influenced by whether mitigation measures may be available and the nature of data with which risk estimates may be refined. The process used to assess risk to bees is consistent with the process used with other taxa in that it is tiered. It consists of an initial screening level, or tier 1, in which conservative estimates of exposure and effects on individual organisms, for example, adult or larval bees, are used to quantify potential risks. The screen focuses on what are considered to be the major routes of exposure, that is, contact and ingestion of residues in diet being pollen and nectar. Depending on whether a tier 1 risk can be mitigated, the risk manager may indicate that the additional data refinements are needed, in which case higher tier assessments are conducted where measured rather than model estimated exposure values may be used. Higher tier evaluations can also include colony level data. EPA is working with its international regulatory partners to ensure the development of exposure and effects tests that can be used to address uncertainties related to the potential effects of pesticides on bees. Since the initial guidance documents were released in 2014, additional acute and oral contact toxicity test methods, that is OECD Test Guideline 246 and 247, with the social non-apis bumblebee, which is a soil nesting species, have been developed. The EPA Office of Pesticide Programs has also participated in ring testing of methods used to test the non-apis solitary mason bee, which is a cavity nesting species. And the agency has been involved with method development for chronic toxicity tests with bumblebee microcolonies. EPA Office of Pesticide Programs has also been engaged in a retrospective analysis of acute and chronic toxicity data on adult and larval honeybees that have been generated or submitted to EPA to determine whether the full suite of toxicity tests is needed or whether certain tests appear to be protective. For example, honeybee oral toxicity tests appear to be as, if not more, sensitive indicators of honeybee's toxicity than acute contact toxicity tests. This type of information enables the agency to better direct toxicity testing and to reduce the number of organisms subjected to toxicity testing. EPA is continuing to rely on honeybees as a surrogate for non-apis bees. In 2017, EPA hosted an international workshop facilitated by the Pollinator Partnership with researchers from academia, government, and industry examining exposure pathways for non-apis bees. That workshop indicated that while honeybees may be reasonable surrogates in terms of exposure through diet and via contact with residues on foliar surfaces, there is uncertainty regarding the extent to which honeybees are protective for exposure through soil or contact with leaf materials. At that time, there were insufficient data to resolve those uncertainties. Where data are available for non-apis bees, EPA considers these data on a case-by-case basis to determine the extent to which honeybees are not suitable surrogates. Data on non-apis bees will continue to be used qualitatively to characterize potential hazard to non-target organisms. EPA is continuing to engage in the development of guidelines for testing non-apis bees to ensure that the suitable methods are available for testing these species and to provide more uniform data with which to evaluate the extent to which honeybees are suitable surrogates. This presentation has provided a brief overview of EPA's process for evaluating the risk of pesticides to bees. I hope that it provides a better understanding of the data used by EPA in this process. If you have any additional questions, my contact information is provided. Thank you for your attention.
Natalie, you're muted.
So many faux pas today. All right, well, that's it. So if you have any questions, go ahead and email Tom. He won't be here in person to answer them, and we'll pass things over to Alessio next.
Okay, great. Let me share the screen. I hope you can see. Sorry. Oops. I hope you can see it. Okay. Great. Okay. So first of all, thank you for the invitation. It doesn't really happen very often for us to have the opportunity to present what we do, especially to students. So that's really great opportunity for us. And also thank you for the nice introduction you've been making. As it was already said, I work for the European Food Safety Authority, and I suspect many of you don't really know what our agency does. So if you don't mind, I would like to spend a few words about it before entering fully into the topic of the pesticide risk assessment for bees. So perhaps most of you know that the main headquarters of the European Union is in Brussels, but there are also more than 40 decentralized agencies scattered across the continent, which are contributing to the implementation of the EU policy. EFSA particularly is located in Parma, Italy, where I'm also talking from. And it is the reference body for the risk assessment for food and feed in the European Union, and basically covers the entire food chain, as we always say, from field to fork. So really every risk associated with the food chain we deal with. Of course, today we'll be talking about pollinators and bees in particular, which is of course an important component of the environment. But most people in the agency work with the risk for citizens, so really for risk to people. And some of the areas covered are pesticides, of course, which is the main topic for today, but also GMO, feed additives, food ingredients, and so on and so forth. That was just to give you a very brief overview of what EFSA does. Now, I don't know how familiar are you with the basic concept of the risk assessment. Perhaps this is a bit too basic, but even if that is the case, just bear with me for a couple of minutes, and I think a refresher will not mind. In our common language, let's say when we talk about risk and hazard, we tend to use these two terms almost in an interchangeable way. But when it comes to risk assessment, these two words actually have a very different meaning. So a hazard is something that has the potential to harm, while the risk is likelihood of a hazard causing a harm. And I've used a very silly example to explain the concept. Imagine that the hazard is represented by fish. In this case, if you have a shark, that's representing a high hazard. But as long as the guy in the upper vignette is standing on the shore dancing and singing, he doesn't get exposed to the hazard. So overall, the risk is low. If, on the other hand, the guy is throwing himself into the water, but the fish is no longer a shark, it's just small fish, then the guy is exposed, but the hazard is low, and therefore you have a pretty low risk of being attacked by this fish. The only situation where the risk is high is where the guy is throwing himself into the water and the shark is there waiting for him. So this is just really a brief reminder to say the risk is always determined by two components. One is the hazard and the other one is the exposure, and both needs to be present in order to have a risk. Now, unfortunately, when it comes to ecotoxicology and in general chemical risk assessment, it's not that easy. And that is because in general, exposure is not something that is there or not there. It's generally a continuous variable, and that could happen at different levels. And the same is true for hazard, which is expressed as a function of the exposure. And that means that you increase the exposure, that determines generally an increase in harm. And that leads us to a very famous quotation, or at least from among toxicologists and ecotoxicologists, from a Swiss physician and alchemist from the 16th century, Paracelsus, who used to say, everything is a poison, nothing is a poison, it is the dose that make the poison. And this is just to say that there is no such thing as toxic chemicals and non-toxic chemicals. It's just really the dose that triggers the negative effect that matters. And risk is always assessed by comparing the hazard, meaning the dose that creates the problem, and the exposure that is likely to occur into the environment. And now entering a bit more into the risk assessment for bees and how it is carried out in the EU, allow me first a bit of historical perspective. The first risk assessment scheme that we had in Europe was produced by EPO, which was named also by Tom before, which is the European and Mediterranean Plant Protection Organization. It dates back to 1992. We started regulated pesticides in 1991 in Europe, so that was immediately following, basically. Another milestone which is important to mention is the 2002 revision of the EPO standard, which was basically taken up entirely by the European Commission, which issued in the same year the guidance document on terrestrial ecotoxicology. And this is the risk assessment scheme which is currently used for assessing the risk on the legal term, let's say. Now you see that after 2018, there was a quite high acceleration of documents which was basically developing on the risk assessment scheme for bees. There was a second revision for the EPO standard, and then EFSA, which was meanwhile founded in 2006, issued first an opinion, a scientific opinion in 2012, and the guidance document in 2013. And today I will be speaking mainly about the risk assessment scheme as it is contained in the EFSA bee guidance document. This document has never really been adopted for the risk assessment in Europe because of the opposition of some member states for several reasons. But I still will be talking about this because it still represents the most up-to-date risk assessment scheme that we have in Europe, and certainly more state-of-the-art than the previous ones, let's say. It has been used, nevertheless, for carrying out some specific risk assessment. For example, the risk assessment for the neonicotinoid substances, a class of insecticide, which we carried out in 2018. This is just for sake of completeness. We are currently working on a revision of the guidance document, which we hope to finish next year, but that will have to be seen. Now, for the development of any risk assessment scheme, at one fundamental step, it is the definition of the protection goals, and specifically the specific protection goals. And these are the definition of what needs to be protected and to what extent. Now, EFSA has developed a method that consists in defining the specific protection goal via 5 dimensions. Actually, it's 5+1, as you will see in a second. These are the ones that you see now on the screen. And basically, for what concern bees, it was decided that the focus should be on the colonies for honeybees and bumblebees, and for population for solitary bees, and specifically the attribute to be protected should be the colony strength or the population abundance. It was also decided that the focus should be on colonies and population living at the edge of the treated field. So not every possible colony that you can have in the landscape, but really those that are likely to be more exposed to the pesticide being used in agriculture. Unfortunately, there was not a clear definition of the temporal scale, but that was interpreted as basically it should be respected at any time, the protection goal. And perhaps the most important let's say, dimension is the magnitude of the acceptable effect, which was defined to be negligible. And later that was translated into an effect which has to be less than 7%. Now, as I was telling you before, you have actually a 6th dimension, which is the exposure assessment goal. Imagine you're in a landscape, your pesticide might be applied to a number of fields. As we've seen before, we're focusing on hives and population at the edge of those treated fields. So in a landscape, you can have a situation like that. The important part here is that every hive or every population at the edge of the field will be experiencing a different exposure. So everyone will get to, well, different exposure level. And this exposure level can actually be interpreted as a sort of a distribution. And the idea for this specific assessment is that every assessment should be focusing on the 90th percentile of the exposure level that you can get in the area of use. And that is basically the exposure assessment goal for the area of use. And it's also the focus of the assessment we are carrying out. So to summarize, once you put together the information from the specific protection goal and the exposure assessment goal, it turns out that the objective overall level of protection is to have less than 7% effect at any time on the strength abundance for 90% of colonies population living at the edge of the treated field in the area of use. Now, the target of the EFSA B guidance document is mainly concerning 3 bee groups. And well, honeybees, bumblebees, and solitary bees. These are not really homogeneous in terms of taxonomy. Honeybees, just one species. Bumblebees, we have 68 species in Europe, which are all belonging to the same genus, which is Bombus, of course. And then we have almost 2,000 species of solitary bees. These are just grouped by that in order to, let's say, have some commonalities, at least in the social behavior. But perhaps this is not a fair let's say, division of the groups, at least not in terms of taxonomies and behaviors. But probably you know about it, about this species more than I do, so I would not spend too much on it. You've already seen that in Tom's presentation. It works pretty much the same way also in Europe. We have a tiered assessment scheme, meaning that we start with the situation where you have very simple data and kind of worst case assumption for what concerns the exposure, rather low ecological realism, and only if a low risk cannot be demonstrated at that step, you basically move up the ladder, achieving a higher ecological realism, but also more complexity in the assessment and more expensive data to be generated. And the main principle is that the more likely is that a certain use of a substance is safe, then less data and assessment are required. Now, remember that risk assessment is always a combination of hazard and exposure. So we'll start with having a look at how data are generated for hazard. We'll be focusing mainly on tier 1 at the beginning. The screening step is just really a tier 1 with very worst case assumption. So it's not really worth looking in depth into it. We'll start with the tier 1 hazard. Well, the list of studies is, you've seen it already from Tom's presentation. We're relying a lot on the OECD standard test guidelines, and that includes acute studies for honeybees, for both oral and contact route of exposure, but that also chronic oral toxicity for both adults and larvae honeybee. There are also standard acute tests for bumblebees, for Bombus terrestris, but these are not a requirement yet. Well, the design of the study is fairly simple. Generally, you start with either young bees and you put them in a cage, as the one in the picture you've seen before, or larvae are kept in grafting cell, as the one you see in this picture now. Generally, you have 3 batches of 10 bees per tested concentration, 5 concentration levels. And then depending on whether you're studying oral or contact route of exposure, when it's oral, the bees are fed with contaminated sugar solution, or if it's contact, they are applied with a drop of the solution containing the substance. There are also validity criteria which are linked to the survival of the colonies, survival of bees in the control, and also toxic standards. So the substances whose toxicity is known and has to be in a certain range in order to prove that the sensitivity of the test setup. At the end of the test, what you do is basically count the number of bees still alive at every dose group, and the maximum dose, which is showing no significant decrease in survival compared to the control, is labeled as the NOED, so the no observed effect dose. And with the rest, you basically draw a plot like the one you see now, where you have survival on the y-axis and the dose on the logarithm scale on the x-axis, and you fit a sigmoidal dose response model to each survival at each dose. And by doing that, you derive the dose corresponding to the expected 50% mortality, and you call that LD50 for acute test or LDD50 for chronic test. Now, perhaps one of the most intriguing aspects about the hazard is the issue of the surrogate species. Remember that the objective for us is to protect all bee species. Nevertheless, the standard toxicity tests are only available for honeybees, and as we've seen, only partially for bumblebees, but again, not a requirement, so we don't have that for every substance. And the sensitivity to the same chemical is likely to be different also in different species. So how to do with that, how to deal with that? And generally what we do is to work out assessment factor to extrapolate from honeybees to all other bees. This was done with an exercise in 2014 by Arena and Scholastra. They've published their results. Basically what they did is to calculate ratio in the endpoint for honeybees and every other bee species. They worked out a kind of a distribution of these ratios, and they've seen that if an assessment factor of 10 is used, basically 95% of the cases are covered. And that's why at the moment, this assessment factor of 10 is applied to the endpoints for honeybees in order to extrapolate to bumblebees and solitary bees. I just want to mention that we are doing another review of the literature for basically every toxicological information we have for pesticides and bees, and we hope to come up with a new analysis soon. Now, moving on to the exposure, well, you already had quite an introduction from Tom on that as well. When it comes to honeybees, there's mainly contact and oral exposure. Contact can be from contaminated surfaces into the environment, and that includes contact in field when the surfaces are oversprayed, or even off field when the surfaces get contaminated due to spray drift or drift from dust particles, that depending on the fact that the pesticide is sprayed, or is it used for, let's say, seed treatment. And when the seed is drilled, then part of it can be drifting away in form of the contaminated dust. Another possible route of contact exposure for honeybees is contaminated wax in hive, but this is not considered currently in the EFSA 2013, partly because the data were not really there, and partly because it was considered to be not really a major route of exposure compared to the others. For what concerned the oral exposure, okay, it was mentioned before, pollen and nectar are perhaps the most important, the most let's say the best studied, and these can happen both in field from the treated crop or for weeds that are growing into the field. And we also have a consideration for the scenario for the successive crop, meaning if you are dealing with a persistent substance, that in principle could persist in the soil and be taken up by the plant the next year and pops up then the next year in pollen and nectar. This is mainly for systemic substances, of course. And then also consideration of the off-field scenarios due to, again, the drift of the substance outside of the field, and that include field margin and the adjacent crop. There is also consideration of the oral uptake for water, and that water can be taken up from guttation, which is an exudation of drops, basically from the xylem sap of the plant, and also puddles that may form into the field, and also off-field when basically there is a collection of water from surface water body that may get contaminated by several processes of movement, let's say, of the chemical from the field to the outside. Finally, there is another possible route of exposure for honeybees is due to the honeydew, but again, that's not considered in the EFSA 2013 guidance document because of lack of data, and again, perhaps not really one of the major route of exposure. As for the hazard, the issue of using a surrogate species is also important also for the exposure. At the moment, in the EFSA guidance document, the exposure routes for bumblebees and solitary bees are considered equal to the one for honeybees, except for water. But in reality, that's not necessarily the case. In order to predict the relevant route of exposure, we should have a very good knowledge of the ecology of the different species. And that's because, of course, behavior, lifestyle, life history traits, and so on are very different among the different bee species. And specifically, the foraging habits and the nesting behavior are likely to influence a lot the exposure to the different life stages. And I just wanted to report here in my opinion, very nice bibliometric analysis, which was recently published in Apidology. And it's really show how the uneven distribution of information we have for the different bee species. Basically 3/4 of the publication are concerning only one species, which is Apis mellifera. Then the remaining part is split almost in 2 halves. One which is concerning only 4 species, which are the managed species. And then for the other almost 2,000 species living in Europe, there is only a small fraction of publication, meaning that we don't know much really for a lot of species. Tom already mentioned this workshop, which was carried out in Washington a few years back, where anyway, there was an attempt to, let's say, try to see whether the exposure of honeybees can cover for the exposure for non-apis bees. I will not spend too much on it because the outcome of the workshop was already presented. Basically, for most route of exposure, Apis mellifera can be considered a good surrogate, but at least for contact exposure from contaminated mud and soil and plant surfaces, this is likely not the case. I just wanted to go a bit more into the way we estimate the exposure at the tier 1. For what concerns contact, that's fairly simple. We just consider basically the application rate in terms of mass of substance applied per area and the fraction of this application rate that is deposited on the target surface. And that's basically considered to be 1 when we're discussing about field scenarios and less than 1 for all field scenarios, but that will depend on the crop and the application used. The exposure estimate for oral is instead, well, a bit more complex, I would say. There is more factors to be included. Again, the application rate is important. There is an exposure factor, which is conceptually similar to the fraction deposited we've seen before, except it also accounts for the way that the substance is applied, whether it's sprayed downwards or upwards or so on. There is a time-weighted average factor, which is only considered applicable to chronic exposure, which basically accounts for the dissipation of the chemical in time. And then we have a so-called shortcut value that estimates the intake of a chemical for a bee for a normalized application rate of 1 kilogram per hectare. And that in turn includes the residues on pollen and nectar normalized for an application rate of 1 kilogram per hectare, the standard consumption of pollen and sugar, and the sugar content and nectar. So we've seen more or less how the hazard and the exposure are, let's say, estimated at tier 1. And we move to the risk characterization, which in principle is just a comparison of the exposure predicted in the environment and the hazard, so the concentration triggering a certain known effect. And this is really just a ratio as it was presented already by Tom. That's the risk quotient. Sometimes it is called ETR or TER, depending on what element is put at the denominator and what element is put at the numerator. But overall, it's conceptually is the same thing. Now, one of the possible interpretation of making a ratio of an LD50 and the predicted exposure is, okay, if you have the predicted exposure is less than the hazard, then the pesticide is likely not killing 50% of the bees in the field. Fair enough. Nevertheless, this interpretation does not account really for the colony and population dynamics, and it also doesn't help addressing whether the protection goal that we had in the beginning have been maintained or not. So in order to make a risk characterization, let's say, consistent with the protection goal, we need to have a sort of reference point. And these reference points are called trigger values that are used to basically compare to the risk quotient. What are the trigger values? They are basically way for translating standard endpoints such as LD50 or LDD50 into effects on colony strength and population abundance, considering the predicted exposure. And the way these are estimated is basically a two-step process. In the step 1, population models are used in order to estimate what is the increase in mortality over time that would hit the SPG, so the decrease of 7% in colony strength and population. And step 2, with the generic dose response, you link that increase in mortality to the standard endpoint, so the LD50 or the LDD50. Now, as we've seen, if the tier 1 is not sufficient, let's say, for addressing the risk and mitigation is not sufficient, then you can move up to the tier 2. In principle, every element of the exposure can be refined, but the most common techniques, let's say, for refining the exposure are concerning refinement of the RUDs, meaning the residue per unit dose, so basically a measurement of the actual residues after the use of the substance. And this can be basically done in two ways. Either you sample pollen and nectar directly from the treated plants, and that represents a kind of a worst case, or you let the bees sample and then you steal the samples from them. And in that case, since you cannot really control the bee foraging behavior, you also account for the so-called landscape dilution. So not all pollen and nectar might come from the treated field, but you still have a good estimate of the exposure that the colony or the population is subject to. Of course, if the second method is used, then the results are species specific because it depends on the foraging strategy of the different species. In general, we say that for avoiding best case situation, we tend to recommend to keep the alternative food sources to the minimal. And in general, we say no bee attractive crop in a radius of 2 kilometers. And in order to have also an idea about the variability of the residues that you can have in the environment, we recommend to have at least 5 independent trials. Another possible way for refining the exposure is to refining the half-life, so the time that is needed for the substance to dissipate. We know this is pretty fast in pollen and nectar, actually, when the substance is sprayed on open flowers. And this half-life is by default considered to be 10 days, which is a rather conservative estimate. So if you want to refine that, the study design is fairly similar to the one that we've just described for RUDs. The only difference is that you need more sample in time, generally at least 5. And once you get the data, you can plot really the concentration versus time, and you can fit the results using a kinetic single first order. Generally, that's the one that is used model. And by that, you define your DT50, your half-life. And with this DT50, you can work out with using the formula that you see on the screen, time-weighted average factor, which is also changing depending on the length of the averaging windows. And that is basically the same exposure window that is used in chronic tests. So 10 days for adult bees and 5 days for larvae. So these are the two most common ways for, let's say, refining the exposure, meaning the tier 2. If still there is an issue highlighted and no possibility from mitigating the risk, there is the possibility to go to tier 3, which are for sure the study with the highest level of ecological realism, but also very high cost and a rather high complexity also when it comes to assessing these studies. The good thing about those is that if the exposure is appropriate, meaning that it is in line with the exposure assessment goal, then the outcome of this study can be immediately used to estimate the risk. There are 3 main types of higher tier studies that are considered in the EFSA bee guidance document, and these are the semi-field tunnel studies, the feeder studies, which are also kind of semi-field in a way, and the field studies. I just want to have a look at each one of them individually and present the principle, the advantages and the disadvantages of all of that. For what concerns semi-field studies, where the bees are basically released into a tunnel, really have a net that is basically hampering the bees for going outside. And the only source of food for the bees is the treated crop, which is generally, and the crop in this case is generally treated before the bees are introduced into the closed environment. And it is often performed with bee attractive crops because basically these crops need to sustain all the bees that are living in there. The advantages is that it requires a limited area per each independent replicate. As you can see in the picture, these tunnels are not massive. They are quite big, but not massive. So you only need a limited amount of area per independent replicate to be carried out. The exposure is a kind of worst case because they have no other choice than feeding on the contaminated crop. While the disadvantages is that the bees, when living in such a closed environment, tend to be stressed, especially honeybees. It cannot last more than a few days, and this is first because the bees are stressed, but secondly also because you cannot prolong it after the flowering period of the crop. Otherwise, the bees will have no food left, and therefore that will create a problem, of course. And for the same reason, you tend to only use rather low, small hives or nests with a rather small amount of bees. Another possibility is to carry out feeder studies. Again, almost a semi-field. Here, the principle is that the bees, both hive or nesting units, are in the proximity of a feeder, which is generally containing contaminated sugar syrup. Sometimes this is coupled with the provision of contaminated pollen pellets. In some cases, the feeder is really within the hive structure, so they really don't have, the bees don't need to leave the hive to feed. And in general, it is performed in absence of significant alternative food source to be sure that the bees are really feeding on what is provided to them. The advantages of this study design is that you have a very good control on the exposure levels. And in some cases, you can even test several doses, meaning that you can, in principle, have dose response relationship at the colony level, which is something that you don't have at the lower tier. And you don't need big areas for the same reason as explained before. The main disadvantage is that this design tends to alter the normal foraging behavior of bees. So also the energetic balance is kind of unnatural. They don't have to leave or to, fly for a very long time to get the food that they need. And that could also mask some sublethal effects that can alter the foraging behavior. Finally, perhaps the highest level of realism that you can achieve is with field studies. And this is basically carrying out by putting hives or nesting units in the proximity of a treated field during the bloom of the crop. The treatment may happen during the bloom or before, depending on the intended use of the substance. And again, it is often performed in absence of significant alternative food source in order to have, let's say, assurance that the bees are actually feeding on the treated crop. The advantage is again, that this is the highest possible level of realism. It's really a very good representation of what may happen in the field after the substance is approved. It is immediately interpreted as a quantification of risk, just looking at the main endpoints, which are colony strength, population abundance, and also has the advantage that the monitoring of the bees can last very long time. In fact, there are examples in the literature of multi-year tests like this. The main disadvantage is that you have, since you have very low control on the setup, then you have many potential confounding factors, so the interpretation is sometimes very difficult. And since you don't have really the control about what bees are doing, the exposure may be substantially less than the goal that you have for your risk assessment. And you can, of course, limit that, but up to a certain extent. And perhaps the main limitation in terms of feasibility is that you really need large areas for the replication. And that is because bees, especially honeybees, can of course fly for several kilometers. And so in order to have really independent sites, you really need large areas for your studies. So this was just to give you basically an overview of all the main tiers of the risk assessment as it is performed. And I hope that was, let's say, enough to give you an idea. And if not, I'll be available then for the discussion. Thank you.
Great. Well, thank you so much, Alessio, for that talk. I thought it was very informational, very interesting. We do have a couple of questions that were submitted in the chat. One was sent in from Wolf, and he wanted to know where the 77% effect number was chosen for the threshold for pesticide hazard.
So how was it chosen?
Yes.
Okay, that's an interesting question indeed. I cannot answer fully how it was chosen. Basically, there was a consideration of what could be the quantification of negligible in terms of being really detectable by a beekeeper. And that was my understanding of, I mean, I wasn't there, so I cannot really tell you the full story, But I think that was the main, let's say, reasoning behind the selection of the 7%, which, by the way, has created quite some problem in reality, because of course it's a very harsh trigger to maintain, and that was also one of the main reasons why the guidance document probably has never been accepted for the risk assessment in Europe, indeed.
That is difficult. And then we have one more question that Christina put into the group chat. And there are two questions, and we don't need to go into too much detail because I want to give Thomas a chance to contribute his thoughts as well in our class discussion. But this question is the lack of data on wild bees due to lack of interest, funding, or challenges in getting enough individuals for appropriate replication? That's the first question. We'll start there.
Okay, well, I don't know whether I'm the best suitable person to answer this question. I would say that honeybees tend to monopolize the attention because in general, they have an economic value that the other bees don't have. And that's perhaps the same also for the other managed bees. So I guess most of it is really due to lack of let's say, willingness to invest on it. But I think this is really changing in the very last year. There's been a lot of interest on wild bees, non-APs wild bees, and I think that there's now many research groups that are working on that. And also as EFSA, we are launching now initiatives in this sense, supporting both, let's say, on a steering level, but also on a economic part of it, projects that are helping collecting more data. Of course, it's really large number of bee species that we have. Only in Europe is almost almost 2,000. So it's difficult that we will get exhaustive data for all of them, but it would be nice to start from somewhere.
Okay, we have a few other questions, but in the interest of time and to get over into the discussion, I'm going to save these, and we can address them in our class discussion as we all hop over to that one. So for those of you who are not in the class, thank you so much for your attendance. We were really happy to have you join us, and you're also welcome to come to our lecture next week as well, which is an industry perspective on IPPM and the framework for that. So thank you very much, and for the students, we'll see you in just a few more seconds. Thank you.